I don't know. Oops, sorry. I don't know if you guys are watching these videos or not, but I'm putting them in there. So you already know about transcription, translation. Is that right? Those are all down packed in here somewhere, so I can ask you a few questions today between lecture and lab. Right, Stephen? Yes? Between lecture and lab. And then it would be nice if you guys read ahead a little bit, look at least the PowerPoints ahead, so you know what I'm talking about. If there is a question, you can ask. Okay, so what happens with this is your ribosome, and in the ribosome, this is the mRNA. And what happens, the mRNA, if the ribosome is floating freely in the cytoplasm of the cell, and what happens, the ribosome can come and attach to endoplasmic reticulum. The blue area, all of the blue area in this model, it is your endoplasmic reticulum. And on this model that I showed you guys from long time ago, you remember this model? A few things about this model. Can you, Ariel, can you see this? A few things. This is your endoplasmic reticulum right here. You remember that? This endoplasmic reticulum connected to the nuclear membrane. Right? And from nuclear membrane, the mRNA comes out to the cytoplasm where endoplasmic reticulum is. And the blue area, you see the blue inside is blue, outside is what color? Light green. Is that right? Light green, whatever it is. So the blue here is the lumen. They call it lumen. Anytime in your body or in a cell you have a tube, inside of that tube is called lumen. Right? Your blood vessels. You have blood vessels. Inside of it is called lumen. Your digestive tract, all the way from your mouth to your anus, it's a lumen of your intestine. So that term you see here, lumen, somewhere in here, he didn't, yeah, ER lumen, endoplasmic reticulum lumen. That means inside of the tube. So what will happen, having said all of that little anatomy of the cell, what happens, this ribosome, which is floating around in the cytoplasm, mRNA is attached to it. It comes and attached to rough endoplasmic reticulum. What mechanism is involved to attach this ribosome to rough endoplasmic reticulum? This is called signal. That's what SRP stands for. SRP stands for signal recognition particles. So that attaches to ribosome. The ribosome, and of course the mRNA, and a little bit of the polypeptide is being formed, signal peptide. They will come and attach to that receptor. Right here is called SPR, binds to the receptor protein. It attaches to the receptor on ER, rough endoplasmic reticulum. We call them endoplasmic reticulum, rough endoplasmic reticulum. It attaches there. Now ribosome is attached on the surface of the ER, and now the remaining of protein synthesis, translation proceeds, and then what happens, I will talk about protein here in a minute. What will happen, this ribosome and mRNA, they all break apart after it's all over. After translation, it's all over. Okay? I don't know how much, I forgot my PowerPoints. I'm sorry about that. I ask you to study the PowerPoints before you come to the lecture, and I don't study them. But quickly, I have to say a few things before I move on. I don't know if they are in the PowerPoints or not. Do you remember that the mRNA, do you remember that? Parts and piece of it break apart. You remember that? And what was that called from last material quiz question today? It was called what? You do it in the kitchen every day. Oh, no. What? Splicing. Very good. Somebody read their notes. Thank you. Splicing. RNA gets spliced, right? Piece of it and bits of it cut off, and the parts that are going to get translated, attached to each other. You remember? They're attached. Those parts that get cut off, what is the name? Huh? The one what? No. You have the cap and the poly-A tail. You have those, but no, I'm talking about right here in the mRNA, a piece of this, a piece of that, of course, when it was in nucleus, got cut off. What those pieces that got cut off and remained in nucleus, what are those pieces called? Anybody? Huh? Introns. Very good. And then the pieces that come out of nucleus, come to cytoplasm, is called what? Exons. Very good. Remember, those are very basic things you have to know. Those are the quiz questions for today. We already talked about it. Same thing happens to protein molecules. All of that protein molecule here is not going to be used by cell. So pieces and bits of it are going to get cut off. That protein molecule right here, a little bit of this here, a little bit of there, a little bit there, a little bit here, are going to get cut off. And those protein molecules are going to be folded. You remember first exam material? We talked about first exam. The protein molecules, I cut up the piece of paper and I twist it and then I fold it. You remember that? First exam material, if you were here during the lecture or not? I don't know. You were late or no. Whatever reason. You remember that? All of that happens in endoplasmic reticulum, rough endoplasmic reticulum, and also Golgi apparatus. So that protein molecule that is made here, this protein molecule is not exact same protein molecule that is being used by cell. For example, I'll give you an example that hopefully make things a little bit more understood. Insulin. We all have insulin. You all heard of insulin. Maricela, you've heard of insulin. Insulin is a protein molecule, if you would, that opens up the door of the cells so glucose molecules can come to the cell, right? You all know that's the function of insulin is. Somebody grab your collar, Stephen, in the mall and say, what is the function of insulin? Just go to the door, say, can I go to the door? I'm not going to run away. Don't shoot me, please. I'm going to, and open up the door and say, that's what the function of insulin is. Insulin opens up the door so glucose molecules come inside of the cell. We all make it. We all make insulin molecules. And the insulin molecules in all of us is same. It cannot be different. Am I making some sense? Even though at the beginning I said the protein molecule in my eye is different than the protein molecules in her eye, different than protein molecules in his eye. Am I making the color of my hair is blonde and then the color of his hair is brunette? Am I making some sense? The color of his hair is blonde and so on and so forth. Those are different protein molecules. But there are some protein molecules in our body that are exactly same. One of them is insulin. The other one is growth hormone. Have you heard of growth hormone? You have a little baby at home growing. Growth hormone of that little baby that is growing is same as the growth hormone at night when you sleep. It's same. All of that is introduction to what I want to say next. So, what happens when your cells make insulin? Before insulin, it is called pre-insulin. Pre-insulin becomes insulin. Even pre-insulin, it was called pre-pro-insulin. Before this, I should draw there, insulin. So pre-pro-insulin becomes pre-insulin, pre-insulin becomes insulin. And where do they get pieces cut off? Those pieces of proteins of insulin, where do they get cut off and being folded correctly Well, insulin does not get folded that much. It's a primary protein molecule in the endoplasmic reticulum. So what am I saying? Bottom line is, I don't know if it's going to come up, but you should know that the protein molecule that cell makes is not exact same protein molecule going to be used then. It modifies. It gets cut off. Just like mRNA. If you forget that concept, remember mRNA goes through splicing. DNA, same thing. It goes through some splicing as well. Not DNA, I'm sorry. Protein. How come you don't stop me and correct me? Stephen, it's your job. It's good. We take YouTube time, Alicia. We take YouTube time. It's okay. YouTube doesn't mind. They're rich. They don't mind. I told you this or no? Did I tell you this? Maricela, did I tell you this? You don't remember. I had a colleague long time ago. We are talking last century. He said, Amir, he was a math instructor, math professor. He said, Amir, I purposely, when I solved the problem, you all had math classes. Brian, you have math, you have physics. Purposely, he makes mistakes during solving the problems. And whoever catches it, extra credit. I said, oh wow, if I do that, after first week, they don't have to come to the class anymore. They all got A. As many mistakes I naturally make, not purposely. He makes those mistakes purposely, and naturally I make those mistakes. I said DNA. It's not DNA. It's a protein molecule that goes through splicing as well. I said DNA. And Stephen didn't catch that. -2 points for you, Stephen. Brian, right? -2 points. He agrees. See, any -2 points I give Stephen, he agrees. He says yes, do it. Let's go. So I spent enough time on this one, and you hopefully we ironed away a few concepts that the protein molecules also get cut off, chop off, until they really become like pre-insulin. I gave you an example. Okay, making multiple bacteria in eukaryotic multiple ribosomes can translate a single mRNA. So you have an mRNA right here. You have, imagine, that's what Einstein said. Einstein said imagination is more important than knowledge. Think about it. But of course you have to have some knowledge too. Without knowledge you don't get far. Imagination is also important. Imagine this is an mRNA. This is a ribosome, this is a ribosome. That's what I'm saying. So before what we talked about, that you had ribosome here and mRNA goes through, tRNA comes in, unload the amino acid, moves, tRNA comes in, unload the amino acid, moves, tRNA comes in. You all remember that? You saw it in the video? Okay, what is saying that that the mRNA can go through a few ribosomes. If you put Maricela's hand here, fist around here, so it could be 3 or 4 or 5, as many, simultaneously forming a polyribosomes or polysome. These are, when I put things in bold, make sure you study them, make sure you know them. Okay, consult your textbook with it. Okay, polyribosomes enable a cell to make many copies of same protein molecules of a polypeptide very quickly. Okay. Yes.
Is this PowerPoint online?
Yes. By mistake.
18 or 19 or?
No, this is 17. Right. 17.
I know, but I was looking at 17. It only goes up to 108 slides. We covered it last time. Is this another 17?
No, continuation of that one.
Yeah?
Yeah. I promise. I did. I modified a few. I cut off a few slides to make you happy. But
Yeah.
is it there?
I'm
If it is not there, let me know. So after lecture, I'll put it. But the thing is, it's not in biology 1. It's on the front. It's on my home page on the front.
Okay.
You see it? Yeah.
Let's see.
Okay, so here is the cartoon picture of it. Ribosome and polypeptide chain. You have polyribosomes right here, polyribosomes. And here is a transmission electron picture of it. The question I asked in last lab practical exam, what equipment this picture has been taken with? You remember that? It was a transmission. There are two types of electron microscope. Scanning electron microscope that scan a cell, okay, and transmission electron microscopes, they cut off the cell and they look inside. That's transmission. I did talk about that at the beginning of the semester. But again, it all depends how much of TLC you do. Lauren, is that right, Lauren? Lauren, do you know what TLC stands for? Huh? No, TLC.
Tender loving care.
Tender loving care. TLC stands for tender loving care. He thought I'm talking something biology.
Yeah, I didn't hear it like that. I think
No, we are too stranded in biology that no, TLC, tender loving care you spend with the PowerPoints, with the lecture material, Alicia, and so on and so forth. But anyhow, these are all of the again, another one, RNA polymerase, of course, and then you have this RNA polymerase right here, and then you have the... Okay, this is pretty much, that's what, Avita, you asked, the cap and poly A, of course, it goes through the introns, these are introns, these are your exons, and then you have the poly A and cap at the end, but this is all exon, right? Even though all of these exon that go through the ribosome, the end product protein molecule, some of these amino acids are going to chop off. They're going to break off. They're not being used for the cell. Am I making some sense, everybody? Okay, so that's pretty much nuts and bolts of it. If you need the whole entire picture in one PowerPoint right here. Okay, and we talked about every step of it in detail, I hope. Right? Alicia, am I making some sense? When I call you names, it doesn't mean just Alicia, I mean everybody. Even Lauren, even Katrina, they're sitting way in the back, enjoying. Huh? No? Okay. When you guys go to movie theater, where do you sit? In the back or front? And why? In the back. Why?
you sit in front, yeah.
She's right. That's why I don't sit up front. In the front, I'm like this. I get a neck ache after the movie's over. Sleeping. Sometimes I fall asleep too in movie. You don't sleep and you don't go to movies and sleep.
No.
I have a brother-in-law. He spent 15, 16 bucks. He sleeps from beginning to the end. He's younger than me, 10 years younger than me, maybe more, 15, 14, I don't know. Younger than me, and he goes to movie theaters and. No, but not Katrina and Lauren. They're not falling asleep. I can see them. They're not falling asleep. Okay, so here again, translation, transcription. Oh, video, I don't know. Some of these videos are okay, some of them are.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected.
Splicing. You remember that? That's what
Finally,
Lumen said.
extra adenine nucleotides are added to the end of the RNA strand. forming a tail.
Poly A,
The completed messenger RNA, mRNA,
Nuclear
now
pore.
leaves the nucleus.
nucleus, membrane. Now translation.
The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA.
Ribosome.
Now, the larger
tRNA.
subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA
Amino
leaves.
acid. Amino acid.
A new tRNA brings another amino acid. The
Amino acid.
ribosome helps to form a new bond, and the process is repeated.
tRNA.
Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs
Anticodon.
with complementary bases on the mRNA.
Codon.
The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids.
Protein. Protein processing. This is
For
only called
proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed.
Very good.
However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane.
Insulin gets
Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
There is a few papers, I think, that when the stress level goes up, students, exams, quizzes, stress level goes up, body craves for sugar. I don't know if you can fight that a little bit. Too much sugar is not good, but that's pretty much what it is. But anyhow, just another one, animation. Let's look at this one.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine.
Methionine, always.
Proteins called
That's the start
initiation
signal.
factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated.
You see the mRNA is shifting, moving.
3 special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
Okay, we spent 40 minutes on stuff that you already know. Let's talk about mutation. What we're supposed to do today.
It was
Yes.
initiation, nomination, and what?
Termination. When the protein release factor comes and sits on the A site. You know, there are 3 sites on ribosomes, E, P, A. The release factor comes and sits on the A site. Okay? Is there any other question? Let's
The release is
to stop the elongation. This is termination. That's what Brian was saying. Termination. Right? That's what the stop signal. There are 3 stop signals that you don't have to know, but you know there are 3 stop codons, I should say. But the start codon, what is the start codon? We all know that. AUG, month of August when school starts. Varicella, is that right? And then what does AUG, what amino acids AUG is responsible for, the codon? Huh? Methionine, MET. So is it true all protein molecules, Amir? Is it true all protein molecules starts with a methionine? Yes. That's a true statement. All protein molecules, but later on they go through splicing and that methionine might be cut off. You remember that? We talked about it. But at the beginning. Let's go to mutation. Any question about mutation? Huh? O'Reilly, where is Trump? Huh?
I think he's on his way.
And he says, I've never been late. I've never been late. I never came late. Okay, that's fine. I know. I have a brain as steel trap. I know. When I used to teach in the forum, you all know what forum is that in Delta College, the forum, South Forum. I never taught in West Forum. West Forum is the biggest one, but in South Forum and the other one, North Forum. You know where that is at Delta College? You don't know. It's a big, huge place. I knew exactly what student is missing and what the trunk thinks I don't know. Anyhow, mutations, one or a few nucleotides can affect protein structure and function. Mutation are changes in the genetic material of a cell or virus. We haven't talked about virus yet. I will later on a little bit, but a cell, when you said a cell, it could be a bacteria as well. A cell, any kind of cell. Prokaryotic cell, eukaryotic cell, a cell. So change in the genetic material. We already talked about this. Is that right? This is the DNA molecule, right? We have A, T, C, G, C, T, A, T, A, C, G. You all remember that? So if one of them changes, one of these guys change right here, and you have C, and the other side you have G. Houston, we got a problem. You've watched Apollo 13 after. I've said it so many times in here. So that's what this is. That's what this is. Originally it's supposed to be TA. But now it's CG. And see what happens. What are the effects? What are the consequences? What is the result? That's what I'm trying to say. Point mutation, change in just one base pair. Change in just one base pair. You guys know what I'm talking about. I didn't write down the other one. T, A, G, C, G, what is it? A, T, C, G. You remember that? This is DNA. I said this is DNA, right? Am I making some sense? So when you change one base pair, it's supposed to be AT. It's supposed to be AT. Now it's CG. That's called point mutation. Point mutation. I hope I'm making some sense. The change of a single nucleotide in DNA template strands can lead to production of abnormal protein molecule. And you know that. We already talked about it, and let's look at some examples today. Here we are. Okay, genetic disorders or hereditary diseases occur. You've heard of sickle cell anemia? I don't know how many of you have not heard of sickle cell anemia. The cells, the red blood cells, look like sickle. They are not nice and round, donut shape. That's what the normal type for here on until end of semester. From now until the end of semester, we refer to it as a wild type, the normal ones. The normal one is called wild type. You're going to go to UOP, Davis, I don't know, wherever later on you're going to go in your life, and you're going to do research, and the researcher will say, this is the wild type. And you will say, oh my God, it's a lion, it's going to come and give you a test tube. They give you a test tube full of bacteria or some other eukaryotic cell or fungi, and say, Maricela, this is the wild type. And you say, my God, what do you mean by wild type? Oh, Amir said wild type means normal. That's what it's supposed to be. That is, the normal one is what? The wild type is AT, right? That's the wild type. The normal one. That's what it's supposed to be. The sickle cell one is the abnormal one, the diseased one, the mutated one, the ninja mutant turtle. This is it, the sickle cell. Okay, so let's look at the sequence of the wild type of the DNA. Are you guys with me? In the DNA, all of this is T, G, C, A. You all know what I'm talking about? All of that. Just these three. Just those three. Let's look at it. C, G, T, A, C, G. And then mRNA is, I guess that he's using the top one. The mRNA is G, A, right? You all know what I'm talking about? Quiz question today. I gave you a piece of DNA. I want the tRNA. I gave you a piece of DNA. I want the mRNA. Am I making some sense? You all know what I'm talking about? Okay. So, and of course the C is G. So this is the template. Right? Am I making some sense? And then of course the amino acids, amino acid here, amino acid for the wild type, the normal red blood cells, the normal red blood cells is glutamic acid, GLU. Stephen, are you ready for the not wild type, the mutated one? So, in the mutant globulin, beta globulin, do not worry about these. I didn't talk about the structure of red blood cells, but don't worry about it. C, A, that got shifted. It's supposed to be T, A, but it is A, T. Am I making some sense? And then of course CG. Now, for this one, for C, mRNA is G. For the DNA is U, right? If it is A, what is it? U. And then for G, for C is G. If you look at that table, the codon table, you know what I'm talking about? The codon table, there are 64 of them. You remember that? If you look at it there, G, U, G, it is valine. Valine, just that one amino acid. Think about it. Just one amino acid make the red blood cells look like this. Just one amino acid. Just one amino acid make the red blood cell look like that, from that to that. Yes?
So mutation occurs when transcription factor doesn't happen. So I remember transcription factor is like the one that determines like what's wrong with it and it changes.
Right. They change it. Originally when DNA, you remember we talked about this, DNA is duplicating, and what happens, or that's one example I gave you. It could be environmental factor, and then what happens, this originally was duplicating, this was placed A, and then said, okay, let's make T here. Am I making some sense? This mistake somehow, some way occurred. Either genetics or environmental factor or something, and of course DNA polymerase goes and fix it, but he does not know to fix it to the original one. All he does, DNA polymerase sees this A and put T in here. Does not put G, does not put C. If it would, then that would be a big chaos. Am I making some sense? I hope I answer your question. I hope. Okay, so that's, and then at the end, glutamic acid is a normal one, and valine is the abnormal one. Types of small-scale mutations, point mutation within a gene, can be divided into two general categories, nucleotide pair substitutions and one or more nucleotide pair insertions or deletions. And I will talk about insertions or deletions in a minute, but nucleotide pair substitution. The substitution, nucleotide pair substitution, replaces one nucleotide and its partner with one another one as a nucleotide. Here is one. I gave you so far so many examples of just one pair. Then just one pair, the silent mutation have no effect on the amino acid produced by codon because of redundancy in the genetic code. For example, UCU, if you look at your codon table, you remember the 64 different type of codon? You remember that, what I'm talking about? The table, if you look at that, UCU and UCG, both of them will give you serine. So it doesn't matter if you change the DNA sequence here, it does not matter to the end product. It's called silent mutation. Okay, both of these will give you serine, amino acid serine. Missense mutation still codes for an amino acids, but not correct amino acids. Does not affect the protein that much. So if one amino acid is misplaced, it's not affecting that much of the protein molecule. In case of sickle cell, it's affecting it a lot. So you cannot say sickle cell is a missense mutation. A missense mutation, it means the amino acid, instead of, for example, proline, it is something else, is isoleucine, does not make that much of a difference where the location of the protein, that amino acids is in the protein molecule. Okay, I hope I'm making some sense. Nonsense mutation, change an amino acid's codon into a stop codon, and nearly always leading to a non-functional protein. The protein is supposed to be this long, for example. This is for example, this time, this is a protein molecule. I know I changed this from mRNA to protein. The protein molecule is supposed to be this long, and then when the cell sees by mutation, sees a stop codon here. Am I making some sense? It makes a stop codon here. Then you don't have the rest of protein molecule. That is an abnormal protein molecule. I hope I'm making some sense a little bit. Okay, so there are different type of single mutation. Just one strand is missing. Here is a wild type mRNA and stop codon. That would be an example of right here, nonsense mutation. I don't know where is the label of it. Right? So stop codon, carboxyl N. And then nucleotide substitution, this is a silent mutation. Okay, so A instead of G. When you have silent mutation, it means either one of these still gives you the amino acid, glycine. Does not matter. Even though they change a little bit, still gives you glycine. And then this is missense, again stop codon, and this is missense right here. Gives you serine. What was missense? Missense does not affect the protein that much. For example, it gives you serine, the serine protein, the serine in that amino acid and glycine, they're pretty much same. They look like same. They're almost acting same in that protein molecule. In that protein molecule. Again, what protein molecule are you talking about? Insertion or deletion. What does that mean? Insertion or deletion. You have, for example, let's talk about insertion. when you insert by mistake, again, this is all mistake, when you're inserting a here, then this whole entire thing got a shift. I hope I'm making some sense. When this thing shifts, then it's called insertion or deletion or frame shift. This is all part of frame shift. This is called insertion or deletion. If you go back to your slide, it says it's a frame. We call it a frame shift. The whole frame shifts. Okay. So what happened? Insertion and deletion are additions or losses of nucleotides pairing gene. The mutation have a disastrous effect on the resulting protein more often than substitution do. Okay, so if you are adding or deleting something here, then the disaster is much more than just one of them change. That's what that sentence is saying. Insertion or deletion of nucleotides may alter the reading frame and producing a frame shift mutation, which I did talk about that. Okay, new mutation and mutagens. Spontaneous mutation can occur during DNA replication, recombination, or repair. Mutagens are physical or chemical agents that cause mutation, like sunlight. Sunlight is a mutagen. Smoking cigarettes, alcohol, these are all mutagens. Okay, so they consider them as a mutagen. The idea of a gene has evolved. What is a gene? Revising the question. That's true. That since I was a student, when I started college, back when none of you were born, the definition of a gene was unit of inheritance. And that's pretty much what I want for this class. But over a period of time, that has changed. The last 20, 30 years, that definition has changed. And I'm talking just my lifetime. Okay. So what is a gene? Revising the question. The idea of a gene has evolved through the history of genetics. What we have considered a gene as, at the beginning, it was described as a unit of inheritance. Over a period of time, that changed. It changed a region of a specific nucleotide sequence in a chromosome. Yeah? Is that it? No, it changed again, and a DNA sequence that codes for a specific polypeptide chain. Probably that's the best definition so far we have. And I know at the beginning of semester I said the gene is a unit of inheritance, but this is the best definition. This is the most current, updated definition that most everybody agrees. A DNA sequence that codes for a specific polypeptide chain. Again, during President Clinton, there was a book or somebody said something, our genes, the question is, are genes responsible for our intelligence? And the person who came up with that got into big, not trouble, but I don't know, how you know how public is, how media is. But anyhow, I'll leave that question for you guys. Hopefully you can get your PhD in it and see if genes are responsible for our intelligence. Maybe not. I'll leave that up to you. I'm not going to. A gene can be defined as a region of DNA that can be expressed to produce a final functional product that is either a polypeptide or an RNA nucleotide. Of course, this is a more elaborated definition of this, and that's why I put it in one polypeptide. Okay, wild type. What are these? A nucleotide pair and one changes. These diagrams, I'm not going to go to it, you guys, but please, you go ahead if you want to understand the material and do well in the class. Go over these, and these are point mutations. All of these are point mutations, and these are the frame shifts. Okay, either insertions occur or deletion occurs, and then the whole frame shifts. So look at it. If I want to go over it, I might put you to sleep, but you look at it, pay attention to it, and come up with these are all frame shifts. These are all, and here is a transcription, translation, putting the chapter into summary. This is summary of everything. The cap, you were wondering about this. You remember the cap and the poly A. The cap is guanine triphosphate, right? You should know that. And the tail to protect the mRNA from enzymes, hydrolytic enzymes, and so on and so forth. Okay. Let's take a 5 minutes break, guys, and then we'll go over next chapter while I'm loading it up. Okay? Get up, stretch. Yes, please get up and stretch. I don't want to talk for 1.5 hours, and you could go get some coffee or drink or whatever you have to do. Very seldom. Should I ask you to get up and do jumping jacks in front of us? No. Okay, great. So please get up and stretch. I don't want you to sleep. Stephen, go run around the block a few times. No, it's
I'll go to the
okay.
gym later.
Huh?
I'll go to the gym later, so
Oh, you go to the gym later? That's good.
I'll do exercise right there.
You go there at 7:30?
No, right after class.
Before class?
Right after class.
Right after. Oh, Stephen.
I think on YouTube.